So once again, good afternoon to all of you. Laboratory animal we will study in today's class. So laboratory animal generally, if we can see, there are different type of animals. So domestic animal, wild animal, and third category we can say that is about a lab animal. So lab animal generally those animals that are generally used in our lab or research purpose, they are considered laboratory animal. So similar to other animals, this animal had also some aspect that need to be studied. So generally handling of this animal and restraining, this is an important one. That will be followed by caging and bedding, just like housing of other domestic animal. Feed water, definitely that's play important role. Beside this one, environmental and physiological parameters, they may vary from species to species, and they are also influencing on their breeding and other physiological parameters. Disease, of course, there are some important disease caused or observed in laboratory animal, and that may leads to loss of that particular colonies of lab animal. So they need to be diagnosed and treated in the time, so that you can spread or you can prevent your particular colony disease free. Then use, of course, our sole purpose is to use them in our research work. So their use might be vary, or we can use them depend on our own objectives or our research objectives. And recently some some area are developing like the proper disposal. So once you will use, you cannot let them to render in society. You need proper disposal of that used animals. So all these things that require to be taken care by some society or some board, and because of this nowadays animal welfare board are enacted under our constitution, and they are responsible for formulating various act and their implementation. And similarly, various international organizations are also working to observe about the welfare of those lab animals. And lastly, there are some international organization, national organization, they are also trying to provide or trying to suggest the use of alternatives to lab animals. So generally they are giving 3R principles or theory. So these 3R's, one that is for replacement. So wherever it is possible that instead of laboratory animal, if we are capable to perform similar experiment in cell culture or on computer based modeling, then it is better to go for those modeling instead of using live animal. Second, reduce. Reduce means if your motto is served by using the less number of animal, then it is better to use the less number of animal. And third, that will indicate that you have to tie refinement. Refinement, that means you have to reduce your procedure so that minimum pain is given to lab animal, so that they can have minimum suffering during your experiment. And nowadays one more R is used, that is reuse. Reuse means those animals, those are already used for one experiment. If the system is different, that may not alter the results of other experiment, then that spent animal can be used for your next experiment, that is reuse. So these are the different aspects, but it is difficult to cover all the aspect in today's class. So what we will, and of course they are not required in microbiology. So they might be, you might be learning in, or you might have learned in LPM department, or you will be learning in your pathology department or other thing. But what is important in today's class, or what I'm going to teach in today's class, that is how to handle or restrain the animals, what is the use of animal, how we can collect the blood, and how we can inject our antigen or our drug. That three parts we will cover. So before starting or going to this one, we should know that to whom we can call the laboratory animal. Generally, these animals are intended to use in research, teaching, or testing. This is just one. And generally, this animal have specialized anatomy. Their genetic and physiological conditions are well known, well studied, and these parameters are already available in public domain. And this particular animal, they might be a wild or domestic, but all the studies, all the data are available in the public domain. That's why they can be used in our research purpose. Beside our research purpose or teaching or testing, some laboratory animal might be used for food production or fiber production. And there are some laboratory animal, because of their look and because of their obedience, they can be used as a pet. Okay, so this is a general. So in brief, if we need to define the laboratory animals, then we can say that these are those animals which have defined health and genetic status, and usually they breed for the purpose to use in research. Means their breeding purpose is only to use them in the research purpose. These animals are generally considered as laboratory animals. Even we can tell domestic animal as a laboratory animal if we can use the domestic animal for our research study. But in general, there are limited number of animals generally used in in research purpose, that we'll see in coming slide. Now, in general, what are the use of laboratory animals? So there are n number of use we can say, but I had narrated few of them. Like we can use this animal for drug or cosmetic development. So that development process that require different type of study, and that study can be performed in this lab animal before performing on actual host. Then second, they may be used for disease diagnosis. So there are many methods, molecular, serological and other methods. But still there are some diseases that can be accurately diagnosed by using laboratory animals. And the promising area of laboratory animals use, that is for production of diagnostic and biologicals. Diagnostics generally require the pure antigen and antibody, or you can say biologicals. Principally biologicals contain the vaccines, or beside vaccine, a pure antigen, antibodies and other formulations. So that are generally in pharmaceutical company, they are produced by using laboratory animals. They can also use for study of pathophysiological of various diseases, generally infectious diseases. Even in genetic study also you can use, and many times they might be used for demonstration purpose to the student for explaining the physiology or anatomy of particular animal. Now, the ultimate aim of all this research, that is advancement of medicine, or you can say healthcare center, so that we can alleviate the suffering of both human as well as animals. So ultimate purpose is to develop some product or some procedure or or some understanding that will help us to reduce the suffering of human and animals. So now with this background, we can say that what are considered as laboratory animals. So I had tried to narrate few in the red and others in the black with etc. etc indicate that beside this list there are many other that we can include as a laboratory animal. These are the one, they are not commonly used but generally used in pharmaceutical companies, and these are the one they are generally considered as a most commonly used laboratory animal. So we will see one by one the characteristic of this rat, mice, hamster, rabbit and guinea pig, and this you have to study by yourself. Now before going to each species, there might be two terms you will come across in your future study. One, that is transgenic animal, and second, that is knockout animal. So transgenic animal and knockout animal, these are the buzzing word nowadays in genetics, or you can say in the era of biotechnology. So these are the type of animal, they are genetically manipulated. Both have similar aspect of modification, but the only difference is transgenic. Trans means you are transferring some gene. Transgenic, that means that you are transferring some foreign genes into those germplasm of native animal, and because of this introduction of new or foreign gene, they are considered as transgenic animal. And this transgenic animal has different roles in production of that particular protein encoded by your foreign gene. In contrast to transgenic animal, knockout animal. So knockout, this is similar to what we are generally hearing the words knockout during your cricket. Okay, so in cricket we are telling that it is knockout round. So knockout means you are removing some particular target gene, so that disruption of that gene leads to the loss of that function. And once the function get lost, you can easily correlate that this function is because of this gene. Or sometimes some harmful gene need to be knocked out so that their effect can be minimized. So that is known as knockout animal. So this difference you must be know transgenic animal and knockout animals. Now we'll see one by one. So first we will start with a mice that is generally named as mus musculus. The use blood collection, how we can handle. So we will see like this, and later on we can say that how we can go for administration of drug, what are the different method, and from where we can collect the blood. And at the end we will see the summary of physiology and other parameters of this important laboratory animal, and then we will conclude our class. So use of this animal, generally mice. Mice is considered as a generally most useful laboratory animal because of their small size and easy to handle. And most of study are generally performed on mice. So mice generally used to perform, used to study the pathogenicity of some viral and bacterial disease. Many times they are also used for production of some biologicals like in hybridoma technology. Sometimes they are also used for virus isolation like in virological study, and many times they are also used for some neutralization study, toxin neutralization or serum neutralization study to demonstrate some toxins or some particular antibody, or many times to demonstrate the presence of antigen. Okay, so these are the brief about brief use. There are many lists, many more lists were there. Okay, now when it comes to blood collection, the most prominent site for blood collection from the mice, that is a tail vein. And there are some organization, they also collect the blood from eye inner canthus. So from the inner canthus of eye, the blood can be collected. So in coming slide I will see that how, I will show you that how we can collect blood from tail and eye, and generally this is handled by tail. So that again I will explain you in coming slide that how we can handle this mice, rat and guinea pig. So this is a second animal that is considered as a rat, and rat is also similar to mice, and almost all the study those we can perform on mice, they may be performed on rat. The only difference between rat and mice physiologically, that you can say the size. I had kept these two photograph so that you can easily compare. This one is a photograph of mice and this one is a rat. Okay, so rat might have different color, or their general body size is larger than mice. So this is an important difference. From seeing you can easily differentiate whether it is a mice or rat. So this thing you should remember, because in exam we may put image in your spotting, and you have to identify which animal is it, and we may ask some couple of questions related to this animal. So this is a difference between rat and mice, even though they are almost similar, but their size is entirely different, and many purpose are also different, study purpose. So generally for blood collection also similar to mice, tail vein is generally used, and many times direct heart puncturing is also performed to collect the large amount of blood. But the handling of rat, that is similar to mice. You can handle the rat by tail and by grasping their ear. Now the third laboratory animal, that is known as hamster. So hamster, that is little bit larger than your rat, and it is easy to differentiate from rat because of that morphology. It looks like a smaller pig, smaller rabbit, and it is very beautiful and shy in nature. Okay. So this particular animal is less used compared to rat and mice, but there are many experiments that required hamster, because that is susceptible to some diseases and some infectious agent. Generally, skin taste and anaphylactic, these reactions, they are generally studied on hamster because of their different physiology that favor the skin taste and anaphylactic shock, anaphylactic study. And that's why there are many cosmetic studies, cosmetic agent studies, they are performed on hamster. Here also we can collect the blood from direct puncture of heart, and the handling of hamster generally that is performed by hand grip around their back, so that fur or back skin can be firmly hold, and then you can hold from your from that neck. Okay, now moving to the fourth one, that is guinea pig or guinea pig, whatever you can pronounce. So guinea pig, that is a miniature of normal pig. Okay, it looks like a pig, but it is very miniature. It has many aspects similar to human physiology, and because of human physiology, that is generally used for human drug trials. In animal, that can also use for disease diagnosis like FMD. In case of FMD, we require to inoculate virus in foot pad, and the foot pad had sufficient space or sufficient area in case of guinea pig. That's why this is considered as an ideal model for study of foot and mouth disease virus. Okay. And generally this is considered as an ideal model for juvenile diabetes study in case of human. Generally, the second peculiarity of this animal, that is, it has a all type of complement. So in your immunology class, you might be study about complement, that what it is, how they are playing the role in immunology, and this is considered as one of the important arsenal of our immune system. So this particular complement, generally they required for performing some test like complement fixation test. So generally this animal is providing or considered as a source of complement, and many times it is referred in formal as a queen of complement. And that's why many times in exam also they may ask that which laboratory animal is ideal for collection of blood sample for harvesting complement. Then definitely there should be the answer must be a guinea pig. Again, blood collection, that is similar to hamster. You can collect the blood by direct heart puncture. Now what we will see, these are the four important animals. Now we are before touching to the rabbit and poultry, because their physiology, their size is little bit different. But all these animals can be handled in similar way. This all the animals can be used for similar type of study, and their physiology and other parameters are somewhat related. So what I did, that I had took all this handling and inoculation and blood harvesting all combinedly. Okay, so even the photograph showing the rat, you can consider this similar for mice and hamster. So the first, that is handling and manual restraint. The first and foremost, that required the correct handling of laboratory animal, so that you can give the minimum pain to the animal, and you can easily handle animal so that that will not harm to handler, and you can easily inoculate or perform your procedure. So for rat, what you have to do, by tail you have to pick up the mice or rat or that animal. You have to take out from that cage. You have to put the animal on the top of that cage. So animal try to move on the front side. So you have to hold the tail, and you have to push little bit backward side. And then from the other hand, you have to grab that animal from the cuff or from between the two ears. Okay, so that will restrict the movement of their head, and because of this restriction, now there is less chances of biting from that animal. Now in the third, you have to reverse. Okay, you have to revert that hand, and you have to put that tail beside your last finger. So you have to hold the tail beneath the little finger, and you have to invert that animal. Now the entire back portion is exposed. Now you can easily go for inoculation for IV or intraperitoneal or for some other procedure. So this is a handling, that how we can handle the mice, rat or hamster. Now this is a restrainer. Sometimes we need to restrain. Sometimes we need to restrain the animal. Okay, so some restrainers are also available. Now this restrainer that required or that requirement is depend on the size of animal, how long you have to restrain that particular animal, and the last that what injection or what sample you have to inoculate by which method. So based on this thing, we can use or we can choose the proper restrainer, and that restrainers are generally available like this. You have to allow that animal to enter inside this one while holding the tail in your one hand. Once it will enter, you can cover them with this flap. So this flap once get covered, now animal get restrained. That will not do any unnecessary movement, and you will have the access to all their body parts. So such restrainers are also available in market. Now, there are different, now once you will restrain, then you have to go for inoculation of your target material. It might be a drug or some antigen or some other way. So there are different methods that depends on your target molecules. There might be an intradermal, intravenous, subcutaneous, intramuscular, intraperitoneal, or there are some study where we can go for intracerebral inoculation. So generally this intracerebral inoculation is generally performed in case of rabies diagnosis. And mice inoculation test is considered as a gold standard test till now for the diagnosis of rabies. Generally albino mice is used, and your suspected material you have to inoculate intracerebrally in that albino mice, and once it will show the disease specific signs after 5 days till 14 days, then that will consider as a 100% accurate for rabies diagnosis. Now we will see some of the important inoculation route. Others are very easy, so need not to go in detail. So this is the first, that is intramuscular injection. So once you have hold this animal, okay, now once that is under control, then from other person may help you, okay. So you may require 2 person to restrain. So that from 2 people, 1 people will restrain the animal and another people will hold that leg and stretch that leg. So that muscle of thigh muscle, you will get the thigh muscle for intramuscular injection. Now this is an intraperitoneal injection. So previous one you can say the scuff is held by this 3 finger, okay. This little finger is used to hold this tail. Then you can say this is entire area is exposed. Now for intraperitoneal, you need not to select this midline. Slightly away from this midline, you have to inoculate your drug, and it must be in such a way that it cannot pierce any organ of abdomen. You can easily feel this when once you will pierce the needle that it is going inside the intraperitoneum. And this route generally used for harvesting of monoclonal antibody. So when you required for bulk harvesting of antibody, monoclonal antibody, this particular route is used for production of these antibodies. Now this is a third, that is foot pad injection. Generally this is performed for some drug discovery, or for some viruses their target site is paired muscle or paired tissues. So this is just plantar surface you have to expose, you have to keep over this instrument, or you can hold it, then you can inoculate your target. Now coming to the blood collection, so generally 2 principal methods can be employed for blood collection from mice and rat or hamster. The first and most common, that is blood collection from tail vein. So the first thing, you have to restrain your animal in restrainer. If you don't have a restrainer, you can simply use a 50 ml tube. You have to insert your mice and just keep the tail outside from your the top. Okay. So now tail is in your hand. On the bottom side, you can see the 2 lateral veins are moving. So these 2 lateral veins, if it is not visible, it is better to rub these veins using Xylol. You can also apply some pressure so that vein get clearly visible. Once it will be visible, you can go for blood collection by insulin needle, or you can also use microhematocrit tube. Now, this will yield, or this will give a less amount of blood. Now when you require some more amount of blood, then you can go for direct heart puncturing, or else you can go for second method, that is collection of blood from orbital sinus. So orbital sinus, that is a eye. So you have to restrain your animal. Now in the medial canthus, you have to enter the microhematocrit tube. So once you will enter the microhematocrit tube, you will slowly need to rotate. So once you will rotate, axially you will rotate and slowly you will pierce. A blood will comes out in that capillary. Once the required amount of blood will comes in your capillary, then you have to take out that tube. You have to put the pressure on that particular area so that bleeding can be stopped. So this is also used for collection of blood in many laboratories. Now, once you will use this laboratory animal, then you have to go for euthanization of that animal, or you can say the proper disposal of that animals. So you cannot let them render in the society. So you have to perform this euthanization. And in general, there are two methods performed for euthanization in laboratory, that is known as humanized killing by carbon dioxide method. Generally, carbon dioxide or chloroform can be used. In this case, carbon dioxide or the chloroform generally placed in an airtight chamber, and a sufficient concentration is raised so that animal get anesthetized. Then that will get unconscious, and after this stage that will get, or there is a cessation of respiration and ultimately leads to the death. So this is a one method of doing euthanasia, that is by employing carbon dioxide or chloroform. The second method of performing euthanasia, that is generally known as cervical dislocation. So cervical dislocation is generally the physical method where you have to grasp the animal on the base of the tail, place it on the small or hard surface without releasing the tail, so that whatever animal body that will get the stretch, and on the neck side, you have to place the pencil or metal rod. So once you will place the pencil or metal rod, then firmly you have to pull the animal backside while keeping your pencil and metal rod on the front side. So that will dislocate the cervical joint, and that will lead to the death of animal. So these are the 2 or 3 methods generally employed for proper disposal of animal. And here I would like to tell you that if it is possible to reuse, then definitely it is advisable to go for reusing of this animal, if that will not alter the result of subsequent test. Now we can see that sometimes that require the identification, because there are large number of animals that may not be easily identified by seeing their morphology, because they are looking one and same. So in such cases, identification can be performed by ear notch or punch. So you can make some fashion of punching, or you can make some number of holes that will help to identify that animal. Many times people are also putting some stain, but stain may get fed. So that will be difficult for long time of identification. So generally ear notch and punching are the common method for identification of laboratory animal. Now we will see the 5th one, that is rabbit. So generally rabbit used for laboratory purpose, and this is generally used for microbial work. Rabbit is generally used for disease diagnosis, skin reaction study, and pathogenicity test. Similar to guinea pig, rabbit is also considered as queen for hyperimmune sera race. So generally question may ask in your exam that if you want to raise the anti-sera, then which laboratory animal is considered as ideal? So this is a particular animal that is considered as a queen for raising of hyperimmune sera, because it is easy to inoculate and that will give the large amount of serum at the end of your experiment. Here also blood collection can be performed from ear vein. They are the prominent one. So small amount of blood can be collected from ear, Whereas if you require the large amount, then you can go for direct heart puncture. These animals are generally handled by ear. Okay, so you have to grasp the ear firmly, then you can handle this animal very easily. Now the last one, that is poultry, or you can say chicken. Chicken were used in different study in different ways. There might be sometimes chickens or fowl has cell use, or sometimes eggs might be used. Sometimes chickens, small baby chickens can be used for isolation of some fibroblastic cell line. So, and generally these animals or poultry, generally they are used to study the poultry viruses like fowl pox, ND, IBD, Marek disease and other things. Okay. The second use, that is for production or for cultivation of viruses. So that will provide the best medium for cultivation of viruses in the bulk. So that may be used for vaccine production. The third, that is they act as a source of blood for performing of HA and HI test. So these are the different use of poultry in microbiology. Now here handling of poultry, that is generally performed by wing and legs. So wings you have to hold, you can lock the wings and you can keep the animal without movement. And there is one word nowadays you might have heard, that is SPF. So SPF generally, that is specific pathogen free. Specific pathogen free means if you are performing some test, then that chick, egg or poultry must be free from that pathogen, so that that will not alter the result of your research. So this is known as SPF, or specific pathogen free animals. This is in brief about the physiological parameters of mouse, rat, hamster, guinea pig and rabbit. Of course, none of this data will be asked in your immunological examination, but this is just to remember or just to compare, and this might be important in your LPM class. And this is the different site of injection. Okay, so this is a species, you can say mouse, rat, hamster, guinea pig, rabbit, bird, and these are the different method of inoculation. What needle you can use, how much amount you can inoculate, okay, which muscle is there, what is the site, this details is given in this table. So at the end of the lecture you can go through this table and you can have some notes about this one. So before concluding this class, the last I had just narrated that this is a buzzing word, that animal welfare board and act, they are regulated at different place, even at the international place they are there. In India, if you can talk, then Prevention of Cruelty to Animal Act was enacted in 1960, and later on Animal Welfare Board was formulated, and under Animal Welfare Board there are different acts were came. So your assignment is, you have to find out that what are the latest board, what are the latest act pertaining to Animal Welfare Board, what committee is working in our institute to get the permission to work on laboratory animal, that you have to find out, and you have to known about their function. Okay, so with this we will end today's class. Okay, so thank you everyone. And before thanks, there are some point I had kept to ponder or to discuss at your level. Okay, so once again, thanks to all for hearing me. If you have any doubt, you are most welcome.